All right, welcome to the Testa Center webinar for academia. So, with us we have Jesper Hedberg, Angela Vaasa, and Albert Mihranyan. Very happy to have you all here. We'll just give you like 1 or 2 minutes to let all the technical issues go out of the way. So we'll wait just 1 or 2 minutes to have everyone on board. So while waiting, I'll explain a little bit about the technical platform. And remove the echo. So there we go. We're using this YouTube platform so that you can watch us over YouTube. And if you're watching from the event page at which you were invited to, then you will also be able to vote and cast your what you would like to say using the Mentimeter tool in the bottom of the page. And we will ask you some questions there. I think there is a question already, and let's see if there is someone who has found it already. Yes, I see there are answers coming in, and the result is something like the question is, how are you today? And it's some of you are super, which is great, and then some of you are great, and two of you are all right, and there's no one saying nah or something like that. So very happy. We're all very happy for this event. So just to ask you some more questions to wait for all more participants, I will ask you yet another question. So there will be like a thing popping up now saying that the presenter has changed the question. And the question is basically, what is it that you do in your work, in your profession, in your academic project, or maybe a title or a project title or something similar? You may answer to that question. So my name is Jens Hammerlund. I'm the head of IT and digital. I do things like helping projects that has a digital angle to their bioprocess experiments. There might be that you would like to use a new app or use a new tool, explore cybersecurity, for instance, in a lab or something similar. I've been with Testa Center together with Jesper since we started, and I would like to moderate this webinar for you. If you have any questions during the webinar, you can put those questions into the YouTube chat that you can see on your right-hand side, and I will present them to the presenters as we go along or in the Q&A after the presentations. With that, I would like to introduce Jesper Hedberg. He's the director of the Testa Center. He's been with the Testa Center since it began, and he'll probably tell you when and so. And he's been performing to that at the GE Healthcare company, which is now the Cytiva company, for 6 years. And before that, I remember he had been talking warmly about his time on the AstraZeneca company in Södertälje, Sweden. And before that, he was an PhD at the Karolinska Institute. And I think that can relate to many of you in the audience. And he also been a student at Uppsala University once upon a time. So without further ado, I'll leave the word to Jesper, and he can get going with introducing himself. If there's something I missed, or get going with the presentation?
Hi everyone. Thanks Jens, and thanks for that introduction, and thanks for stating that I studied chemistry in Uppsala once upon a time. It's like a long time ago. Perfectly fine. It was a long time ago. I have some 20 years in the industry since then. All right, so it's a great pleasure to present the Testa Center to you guys, and together with Angela and Albert Mihranyan here during this webinar. I'll show a couple of slides around Testa Center. I'll say something about the background. I'll say something about the instrumentation that we have here in Testa Center, and of course how we run projects here in Testa Center. And then we'll also leave it to Angela and Albert to present the projects that they have performed in Testa Center as examples of what you can do in Testa Center. So with that, I'll start my presentation. And if I can get a thumbs up from Jens, if you see my presentation, very good. Thank you. So Testa Center is a test bed, or a research facility, if you like, around both biological and technological innovations for production of biological products. We have a vision. The vision is that we together, and with the word together, we mean that with you guys, with us, and with the projects being in Testa Center, represented here by Angela and Albert, we together accelerate the journey of world-changing innovations. We do this by enabling researchers and companies to verify their innovation in an environment which is relevant, meaning the environment that I'm standing in now, which basically is a production environment. And by being in a relevant environment, we also accelerate the industrialization of these innovations. Also very important, we have a third pillar, secure excellence. So the development of biological production has been very, very fast the last 10 years. So we're lagging a little bit in competence, both as nation, but also globally, I would say, in our educations for students, but also in research community around biological productions. Okay, so Testa Center is a joint initiative between Cytiva and the Swedish government to support the growth of life science. And in order to be able to receive lots of soft funding that we did from the Swedish government, we created a separate legal entity that we defined as non-profit. So the intention of Testa Center is not to generate cash. Its intention is to enable researchers to come to this open access lab and do experiments to accelerate innovations. We are open both internationally and nationally for academia, of course, so research groups doing either basic research or applied research, and also educations. But then we're also open for smaller and bigger companies. We're open for biological innovations, being typically a new protein that you want to scale up the process for, or perhaps a new way of expressing a protein, or something like that. But we're also open for technological innovation, so typically a sensor that could make the bioprocessing more efficient, or digital applications and digital tools to make the processes go more efficient than before. We describe ourselves as an industrial infrastructure lab, so that means then that it should mimic a industry, a production industry for biological production. We are, however, non-GMP, meaning that you cannot generate material for clinical trials here. So the intention is to do scale-up experiments and to reduce risk when you go up in scale. We're also open for education, as I said. So Uppsala University are performing several student exercises here in Testa Center. So right now, actually, in the lab next door here, we have 5 students doing experiments on a bioprocessing start-to-finish process. So in 5-liter scale, including culturing, harvest, and also protein purification. The offering that we have here in Testa Center is relatively simple. The intention is that research groups come here to Testa Center and get access to the instrumentation that we have here, and also the bioprocessing lab. So that's a very tangible thing that they get access to. What they also get access to is that we help to plan the experiments prior to coming here, and then we also provide technical support when being here in Testa Center. Not everyone are comfortable with running, for instance, a 50-liter bioreactor that we see here behind me. So we need to support many of the projects coming here. What we do not provide, we're not the contracting lab. So in that sense, we do not take on assignments, meaning that someone sends a sample here, we purify it, and then we send a purified protein back, for instance. So we're not the contracting lab in that sense. And we do not provide chemicals and consumables for the specific project, so you need to bring that yourself. So chemical, consumables, and resources in the lab. Apart from that, it should be a relatively all-inclusive experience. Of course, all the small things, nitty-gritty, that you need in the lab, like pipettes, scales, and stuff like that, we have. But also get a meeting room, Wi-Fi, printer, and since we're on the site of Cytiva here in Uppsala, we also piggyback on the facility service that is available here. So security, waste management, what else? Printer service, shipment, for instance, and stuff like that. So as I said, the intention is that projects can come here and they can grow and they can de-risk. And what I'm trying to illustrate here with this picture is that you need to be in the Testa Center. But while in Testa Center, we have lots of opportunities. Very, very often when projects come in here, they have an idea of how to drive their project through the execution phase here in Testa Center. But along the way, things often happen. And as you see, I hope you see, most of our instrumentation is on wheels, meaning that we are super flexible with the instrumentation that we have here. So we can roll in and out instrumentation depending on project needs. Also, we have the bioprocessing experts here in Testa Center. So while doing your experiments in Testa Center, you have a bioprocessing expert side by side to coach you and help you if you run into troubles. These are some of the feedback from the projects that have been in Testa Center that we have collected. So lots of projects need to show scalability of their process. They want to go from a small scale process, for instance, or perhaps in a shake flask scale, and then go to a bioreactor scale. Or they already are in a bioreactor scale and want to go even further and make the process scalable. Many of the lab scale processes are not really scalable, so you need often to transfer the initial lab scale processes to a scalable process in a bioprocessing lab like this. A very typical example is going from batch centrifugation, which you usually have in a small shake flask scale, and go that to, so the harvest is a batch centrifugation for that scale, but batch centrifugation is not really scalable, so you need to switch often to either continuous centrifugation or a filtration method. Manufacturability is also important for many. So showing that the molecule, the candidate molecule that you are working on actually works to produce in larger scale. It doesn't precipitate, it doesn't fragment. It has the quality attributes that you previously have verified in the small scale. And doing that in large scale is also important when going forward. Many of the projects want to show robustness of the processes in a semi-large scale. So that's important also for some. A big thing for many is to build know-how and to grow your team when you're here. So knowing your process when going forward with your project. So perhaps you want to engage with a other partner that has a larger scale capacity. Perhaps you want to contract something out eventually. So knowing the process in those instances is very important. Many academics also want to generate more material for whatever reason, either to do their research on, perhaps they have a small scale process that they need to run several times to generate the amount of material that they need for one experiment, or they want to do new types of experiment that needs more material. For instance, we've had projects in Testa Center that's going to do neutron scattering experiments where you need 100-fold more protein to generate crystals, for instance. Another thing which often we get feedback on is the flexibility of instrumentation that I said. So we can save time by rolling in a new type of equipment when needed during the project execution. Just a very brief information for the Swedish companies and the Swedish academic groups that has a commercialization focus. There are funding to apply. So please contact me or Jens if you want to know more about this. Up to 800,000 Swedish crowns there are funding to be received from Vinnova. This is the instrumentation list. It's a busy slide, so the intention is not to go through all the details. Please, you can download the instrumentation list from our homepage if you want to go into the nitty-gritty details. But very, very briefly, we have the cell culturing capacity for mammalian and microbial cells, all the way from a small scale, of course, the shaker incubators to do the seed train. The first bioreactor step in, say, 1 to 5 liter scale. We have the XDR50 for 50 liter scale, and then for mammalian cells, we have a 500 liter bioreactor as well. For the microbial capacity, we're going to install a 200 liter bioreactor in May. So in next week, actually this lab will be rebuilt with a 200 liter bioreactor to stay here. And then we have the harvest equipment to match that scale, so either a filtration procedure or a continuous centrifugation procedure. Of course, we have the batch centrifuge also, if that is applicable. Cytiva is by heritage a protein purification company, so I think it's safe to say that we basically have any instrumentation that you need around protein purification. So everything from the ECTA Start, which is the smallest one, ECTA Pure, ECTA Pilot, which is just beside me here, and all the way up to a full-fledged ECTA process system. When it comes to analytics, we're actually quite basic. We have the analytics that you need to be able to run a process, which means cell counters, metabolite analyzers for upstream, microscope, and we actually have a low voltage electron microscope as well from Vinnova. And protein analytics, again, the basic SDS page, HPLC from Agilent, Gyro Lab workstation from Gyros, and you can do Westerns as well, of course. We do have a BSL-2 lab, not in this particular one that I'm standing in right now, but the lab next door here. So that lab is equipped with airlocks and with ventilation and security procedures to suffice a BSL-2 capacity. So should I say the mild viruses like influenza, rota can be cultured there, and we can also run gene therapy processes like adeno and adeno-associated viruses processes. Typically, again, a busy slide, but just to give you an idea of how a project enters Testa Center and then exits Testa Center. It always starts with a get-to-know meeting where basically we introduce each other to each other, and very, very often we start to discuss the potential project that are coming. We usually do a feasibility, a very, very initial feasibility on these first meetings. Is it possible to drive a project like the proposed one at all in Testa Center? Sometimes there are stoppers like we cannot harbor, for instance, radioactive compounds. We cannot culture all the organisms in the world. We need to have the environmental permits in place and so forth. Very often the discussion goes onward to a planning and prepare period. So this planning and prepare period is usually between, I say the shortest one is actually 2 months or something like that. And the longest one has been for more than a year. So depending on how mature the project is and how ready you are to enter Testa Center, the planning period can be shorter or longer. Very important to say that we from Testa Center is always a part in this planning period. As a someone coming outside of Testa Center, you typically don't have the intimate knowledge around the bioreactor XDR50, for instance, but we do. So we can provide that information to you as a project. What do you need to think about? What do you need? What consumables are needed? How to monitor the process and how to plan the experiment. So we support the projects entering Testa Center in doing those kind of plans. And then when you come to Testa Center, of course, there's an execution phase where you as a researcher need to be here, but then we'll be here side by side with you to support you through the way.
So Jesper, I would like to tell the audience that it seems like the questioning chat for YouTube was a bit hard to find, but now there is also in the Mentimeter a possibility to ask Jesper questions. So please ask Jesper any questions that you have in the Mentimeter, and I'll take them up with Jesper in the end of the session. And you have about like 5 minutes left.
Yes, perfect. So I'm closing in. A very, very short slide on one of the projects. We have Angela and Albert to present a little bit more extensively. Just a brief teaser. Professor Suparna Sanyal has been in Testa Center and done a scale-up of her process that was in the milliliter scale in shake flasks to liter scale in bioreactors, and increased her yield and material from 80,000 picomoles to 300,000 picomoles. So more than, what's that? Threefold at least. It's a several-fold increase, but most importantly, she did a lot of time savings. So from a 7-day procedure, she went to a 12-hour procedure. So that's really fun to see her gains in that. You have another example with HO cell culture, where Sara Mangsbo, also associate professor at Uppsala University, she did a proof of concept for expressing one of her drug candidates, going from shake flasks to a stable expression in a bioreactor scale. This was a mammalian process and took her a little bit longer than typically the microbial processes. They stayed here for 10 weeks in Testa Center and gained knowledge around both upstream and downstream processes on how to culture these cells and how to purify the candidate drug. But I'll leave it to Angela and Albert to show more examples. With that, the very short summary of Testa Center. So we're non-profit. We'll give you expert support both in the planning phase and when you're here. We are non-GMP for biological production, and we're a private-public partnership between the Swedish government and Cytiva. You get access to a unique equipment park that we have here in Testa Center. You get access to our expertise. You pay per the usage, the time unit that you are here, and you get a professional network while being here. And what many projects achieve when they come here is that they reduce the risk when they go up in scale. So reduce the technical risk, start to tick those boxes for this risk has been mitigated. They also increase the process know-how significantly when they are here. I hope that Albert and Angela will share that experience with you. And by increasing the know-how, they also increase the value of their projects. I haven't said anything about results and IP, so let me finish off with that. The results and IP that you as a client generate here in Testa Center is yours alone and yours to take with you. So no strings attached to Cytiva. You don't need to show your results to anyone. You need to actually, for security reasons, I need to know what you're doing in the lab, but I'll be happy to sign a CDA with you so that you can perform a project under confidentiality in Testa Center. With that, these are some of the companies, or actually all the companies that have been here. So shortly we'll have Angela from Solis BioDyne and Albert Mihranyan from Uppsala University to present more. But I think it's time for if we have any more questions. Jens.
Yes, so let's see. I encourage you to ask questions and still continue to write them, if even though I'm picking them up now. They will come into the question system as we go. So Jesper, this is a question I've heard many times as the first question that pops up to many, and it is, how much does it cost? And
Yes.
then linked to that is actually another question that I will take in the same sentence, which is, please link to the Vinnova funding. If you can explain a little bit about that. So those two together, perhaps.
Yes. So first off, included in the cost, you pay for the service when you do the execution phase here, which means that if you plan to be here for, say, 4 weeks to do lab work, that's when you pay to be here. But included in that cost is actually the planning phase as well. So the planning phase, we don't charge by the hour under the planning phase. If we were independent contractors, we would, but we're not. We're a private-public partnership, and we're an open access lab. So we do this planning phase together, and we support you the best we can in that. So included in the whole access fee for those weeks that you're here is also the planning phase and the competence that you can, the help with the competence that we have. But for the actual execution phase, for an academic project, meaning that it's state aid ruled as non-economical, we can explain more, but a academic group is typically defined as a non-economical. It's 80,000 Swedish crowns per week. Now, that's a lot of money. And it's the reason for that is that we're in a production environment. So production environments are relatively expensive to run. We do have some fundings, both for academics and companies, to apply to help you with the financing situation for that. The finance situation or the funding that you can apply for today is up to 800,000 Swedish crowns. You need to provide a 30% co-funding with the project budget, which means that the project budget of 1.1 million Swedish crowns, for a project like that, you can get 800 Swedish crowns to support you. Also important to say, in that budget, you can include several things. You can include the access fee for Testa Center. You can include the labor cost of the individuals being in Testa Center performing the project, and you can also include the consumables needed in project. So it's the total budget that you can get funding for, which then is, I think, is a good way of lowering the threshold of coming to Testa Center. I hope that answered the question.
I think so. Thank you very much. And let's see, we have not much time, but let's have a short question. What if I don't have the particular expertise for a particular instrument that I would like to use for my project?
Yes, so very good. I didn't touch upon that. So not everyone are super familiar with running a 50-liter bioreactor. So perhaps you need support with that. Again, we as a Testa Center organization do not per se provide that support by the hour. We do, however, have many contacts in the municipality and the region around for independent contractors. So if you have the need for your project, someone to actually be dedicated to run this bioreactor, you don't have that in your project, you need to hire that kind of competence. We can help you. So we from Testa Center, and also I didn't mention that, but we also have a very close connection with Stuns. It's a foundation here in Uppsala with the aim of aim of bridging the gap from academia to society to industry. So Stuns has also a very good network of contacts around the independent contractors that can help your project.
Thank you, Jesper. So with that, I'll take you off the spotlight for a while, and I'll ask Albert to prepare, and I have your presentation here, Albert. So let's see, I have Albert, you sent me the presentation for me to present, so I'll fix that. But before that, I'll just give you a bit of introduction. As I understand it, you're professor of nanotechnology, and you're program coordinator and responsible for the energy technology at Uppsala University, where you have been about 9 years, if I understand it correctly. Now, if you have anything to add to your presentation, like your introduction here, I would like you to do that. And I'll, in the background, get going with arranging your presentation.
Yeah, okay. Just to add, I've been at Uppsala University for more than 20 years. I'm a professor in nanotechnology working with nanomaterials, mainly nanomaterials for life sciences applications, but I've had also projects in energy storage in the past. Maybe we can talk about it some other time. Yeah, so let's wait for the presentation.
All right, so let's see, it's coming up. Sorry about this, it's a bit unprepared, but we'll soon be there. All right, so we can see the presentation now.
Jens, it looked like Albert froze, at least for me. The same for you?
No, I can see Albert fine. Thank you. So.
And you're on mute, Jens.
I can see Albert fine. It seems like he's ready to go, and the presentation is on the screen for the audience to see on the webinar. So with that, Albert, you need to tell me when to change your slide.
Yeah.
We're on slide 1.
Yeah, I don't see the presentation, so I will open myself, and then we can take it from there. All right. Hello, everyone. Very happy to talk about our experience at Testa Center and what we have done there. Next slide, please.
We're on the second slide.
So our project was a part of an EIT Health project called VIREPAP. EIT Health is standing for European Institute of Innovation and Technology, which is under the European Commission. And the consortium for our project was Uppsala University, the pharmaceutical company Bayer, and also a small startup company from my research group. And then we also had project partners, Testa Center, Charles River, and CSL Behring at different stages. So what the project was about was to test a new type of filter for bioprocessing applications. And it was to verify the technical feasibility both in lab and on pilot scale. And this is when Testa Center was really important for us. Next slide, please.
Yep.
So our filter is a filter that removes viruses. And first, maybe one needs to explain why virus safety in the production of biologicals is such an important issue. Next slide, please.
Yep.
Last year, I think at around this time, there was this publication coming out in Nature Biotechnology, where essentially largest biopharmaceutical company had written a joint article where they have highlighted the issues with the viral contamination in biological manufacture and the implications for emerging therapies, and that was mainly for cell and gene therapies. But the issue of virus contamination was already highlighted previously in the plasma industry and also in the manufacture of recombinant proteins. Next slide, please.
Yes.
So in this part of the project, because we looked both at the upstream bioprocessing and downstream bioprocessing, in this part of the project where we have been working with Testa Center, we focused on viral safety in the upstream, and that is the cell culture and the contamination of the media that are used to culture cells. And one particular reason for focusing on the upstream is that the bioreactor provides the excellent environment for replicating all these potential contaminants. And which are the contaminants in In the cell media, this could be bacteria, this could be mycoplasma or viruses. Normally, all these components are filtered through sterile filters or treated in some way. But sterile filters that have a cutoff of 0.2 micron, they do not remove viruses. And they have very poor clearance also on mycoplasma. And because cells cannot be sterilized, you can only ensure that all raw materials that are entering the bioreactor, they should be contaminant-free. This is easier said than done. Next slide, please. So there are some really famous cases documented from the past where virus contamination has caused really huge problems to the industry. I should say that virus contamination is extremely rare event, but when it happens, I mean, the consequences are catastrophic. And probably one good example is what happened at the company Genzyme in 2009. In 2008, this company was making $2.1 billion on two products, Fabrazyme and Cerezyme, and everything was looking fantastic until when in 2009 they discovered that they have a virus contamination in their bioreactor by a virus known as vesivirus. This is a virus that is about 40 nanometer in size, related to canine type of viruses or having source in dogs. And due to this virus, the entire manufacturing facility had to be closed down. All equipment needed to be disinfected. The processes needed to be revalidated. There was about 8,000 people who were highly dependent on this drug, who were left in limbo when these supplies of the drug could be restarted. So it is estimated that this single incident of virus contamination at Genzyme facility in Massachusetts in 2009 cost about $200 to $300 million. And when a similar incident happened a few months later at the site in Belgium, this put really the company on the brink of bankruptcy. And what happened was that another company, Sanofi, just bought Genzyme and took over the company completely. And it's still not clear what was the reason for the virus contamination, but it's believed that the raw materials were the most likely source of it. There was another case of virus contamination with another type of virus that is called Minus virus of mice. This is a parvovirus, a smaller virus, which is very difficult to disinfect or remove. And this was at the company called Merrimack Pharmaceuticals, also in Massachusetts. What is interesting about these cases is that normally when you have a virus contamination, it's quite often due to animal-derived raw materials that are used. But in this case, there was no animal-derived raw material that was used in the production at all. Yet virus contamination of the bioreactors has occurred. The financial losses were not as dramatic as in the case with Genzyme. However, this example showed that virus contamination can happen also when you use completely non-animal derived raw materials. There is a theory that the contamination was due to some rodents and hydrolysats from agricultural sources that were used as supplements in the cell culture. So these rodents have been around either in the fields or at the storage location, and then even the slightest contaminant could replicate in the bioreactor. So what are the potential contaminants? As I mentioned, the most common cause of virus contaminants are animal-derived raw materials. Bovine serum is a source of many types of potential virus contaminants, including bovine viral diarrhea virus, Kashi Valley virus, reovirus, and so on. Trypsin, that is derived from pigs, could be source of porcine circoviruses or parvoviruses. There are a number of human-derived raw materials that are also used in bioprocessing. This could be human serum albumin or transferrin. There is a number of viral contaminants that could be from humans. And as I mentioned earlier, there are also environmental contaminants, which can come from rodents, from cats, from dogs, and so on. So, of course, there is a risk. It's a rare event, but there is a risk with strong, very, very big consequences. So what are the risk factors of virus contamination? It's the very low virus titers normally when these raw materials come in. And it's also the inhomogeneous distribution of the viruses in the raw materials. So it's very difficult to detect. Quite often there is little or no visual cytopathic effect in the bioreactor, and there can be a long lag period before the detection of the virus if such detection is based on cytopathic effects. So to prevent virus contamination, active steps must be taken. These are screening of materials and suppliers, use of chemically defined media, and use of different sequencing assays that can speed up the detection. But still, if all these measures are prevented, there is always the risk of virus contamination. The typical methods of virus clearance of cell culture media include heat, pasteurization, high temperature, short time. However, this is not suitable for a number of heat-sensitive components. This also requires the presence of stabilizers, such is the case with, for example, human serum albumin. UVC can be used. However, it's difficult to implement for large volumes. It would require low flows and low penetration depth. And there are a number of resistant viruses to UVC. Gamma irradiation is another way of ensuring virus safety. However, it's also difficult to implement for large volumes. It's not a point of use process. You need to send your raw materials to another site, and it also requires large capital investments. And then lastly is the virus filtration. And what is nice about virus filtration compared to the other methods, it really removes all contaminants if it's based on size. So it removes everything that you know could potentially be in your product, but also things that you have no idea because it's based on size. It's extremely robust. However, the price of virus filters is very high. And this is a big limitation for upstream bioprocessing. So with that in mind, a few years ago, we have developed a filter paper, the world's first virus removal filter paper, that consists of 100% cellulose nanofibers. And that is capable of removing all types of viruses from protein solutions, cell culture media, buffers, essentially as easily as brewing coffee. As one of our partners says, this is the Melita filter on steroids. And what is special about the filter is that we can control the pore size distribution on nano level with extremely high precision. So we can really tailor it to specific particle size fractions. Before we went to Testa Center, we had a number of studies specifically addressing the virus contamination in upstream bioprocessing. So we have done studies, for example, on fetal bovine serum, which is the most common supplement for cell culture, especially on small scale. However, FBS is a very tricky material. Actually, it's poorly defined. There is high variability. there are a lot of fraudulent batches. I mean, the volumes that are reported to be produced and then sold quite often are mismatching. And there is limited supply capacity nowadays with the increase in the demand for novel cell and gene therapy products. The need for FPS has increased, and there are only 3 companies in the world today that are essentially supplying 90% of the market for GMP production for FPS. And being animal-derived raw material, there is always a high risk of virus contamination. Surprisingly, even though there are chemically defined media nowadays, 80% of by triple filtration through 0.1 micron filters. And these would remove mycoplasma and bacteria, but it would not be super effective against many viruses, especially the small-sized viruses. There is something that is called defined FPS, which additionally would be processed through 40 nanometer filters. But these would still leave the small-sized viruses as potential risk. And then to cover up for the risk of potential contamination, FPS may additionally be heat inactivated at 56 degrees for 30 minutes or gamma irradiated. So what we wanted to do was, yeah.
May I interrupt to ask the audience to remember to, if there is any questions for Albert, please remember to fill them into the Mentimeter so that I can forward them in the end of the session, as we're moving towards the end of the time for this. Well, the time is about 5 or 10 minutes.
Yeah. So what we have done, we have looked if we can tailor make our filter to 20 nanometer to remove all types of viruses, and then looked at the cell growth of CHO and hex cells. And we indeed saw very interesting phenomena. There was quite a lot of loss of protein because serum contains a lot of different protein sizes. But surprisingly, the 20 nanometer filtered FPS could very nicely support the growth of CHO and hex cells. And we could show more than 4 log virus clearance, that is more than 99.99%. And with these data in mind, we went to Testa Center, and then we wanted to scale up the process because it's a regulatory demand whenever using filters or developing filters, that these filters should perform on small scale as well as on the large scale. And even though the large scale, what we call a large scale or pilot scale, is not really the largest scale imaginable for these kind of filters, it was still a very big step forward for us to go from growing cells in small flasks to growing them in wave bioreactors and doing all these things. And we have used some of the chemically defined media that were available through Cytiva, with all kinds of cell supplements. And we used a special line of mAb producing CHO cells. And we looked at a lot of parameters. It's the analytical instrumentation that Jesper mentioned to you. We looked at cell viability, cell growth rate, density, a lot of different assays for metabolic activity. Also the mAb yield and the virus clearance. And when we did the filtration for larger volumes, for 5 to 10 liters at the time, we could see that we have a very stable flux. There were some protein aggregates that were nicely removed by our process, and we had very high virus clearance even when we had large load volumes per unit area of the filter. What we did also with the cell culture, we compared the data that we would get from the wave bioreactors with the standard that is the stir tank bioreactors, and the data that was already available at Testa Center to see how different the cell growth would be after the nanofiltration compared to the stirred tank bioreactor. And what we could see, and this is I think no secret, that the wave bioreactor is a lot milder on the cells, so there was less of cell membrane damage, and we had very high cell viability values, although the cell density was slightly lower. But these differences are mainly due to the type of the bioreactor used. And then the most metabolic activity parameters for both of the reference and the pilot system were comparable and within the same range. And the same can be said about the yield of the monoclonal antibody. It was slightly higher for the stirred tank bioreactor, but when normalized per number of the viable cells, these values were essentially the same. So in conclusion, I want to say that the study at Testa Center was very important for us to really show that our filter works not only on the lab scale, but also on a larger scale, on pilot scale. And whatever differences that we observed in the outcome were not related to our filtration process, but were due to the type of the bioreactor that was used. And we showed that we have a suitable prototype for point-of-use virus removal bioprocessing that could be useful for many groups or companies that could be interested in small-scale cell culture for developing next-generation cell and gene therapies. I want to thank all my colleagues and all people at Testa Center who have been really involved from day one when we came to Testa Center and really helped us with every single detail of planning and then executing the work. I also want to thank our funders at EIT Health and our partners at Bayer, with whom we did a lot of similar studies on downstream processing. The EIT Health office and then the Uppsala University Innovation. And lastly, I just wanted to mention about my conflict of interest. I am the inventor behind the IP for this virus removal filter paper, and the company that I mentioned, the SME, is a spin-off from my research group. Thank you very much.
Thank you, Albert. The digital applause. I'll just take away your presentation for now and get back to the Teams meeting. Great. And I'll look up the questions that had arrived. So let's see. Oh, there was actually some questions of maybe we forgot to turn slides after a while. Sorry about that. That might be the case. So then the questions that have come in have been, how many can you be inside the Testa Center lab? I guess this is a COVID question.
Yeah.
That's maybe a question for Jesper.
Yes, so as you see, the lab is relatively large. It's actually one of these labs. We have 4 labs in the house. Maybe I didn't mention that, but this is the medium-sized lab. It's roughly 100 square meters in size. We have, at the moment during the pandemic, we have a restriction of maximum 8 individuals per room. So this goes for this lab as well.
And then there are some questions here that is for Albert, I'm pretty sure. What research environments were you evaluating for your project?
So for us was quite important to come and get access to real industrial equipment. So our aim was to test the feasibility of our platform on different scales using real life samples and in real life industrial environment. So no matter how much we would be doing things at Uppsala University or in my lab, so I think the access to the industrial kind of equipment and then being in this environment was really a big add-on to our project. And then was also access to some of the special special materials. So we were very lucky to get access to some of the mAb producing CHO cells, which were not our cell lines. This was a really kind gift from Cytiva to enable our project. So I would say the access to industrial equipment would be the biggest add-on. Wave bioreactors, pumps, all kinds of things.
Right. Then there was someone who sent in an information that Uppsala University has 25 weeks available, eliminating the need for access fee for research and education from Uppsala University, but there's an application needed. And thank you. And then we had
comment on that too, that we are also very thankful for some of the weeks that we got at Testa Center through Uppsala University, and it was a joint effort. So part of it was funded through our project, and then part of it was funded through Uppsala University.
So last question. We'll see if we can keep it short, but what was the hardest part of your project?
I think it all boils down to very good preparation. I think you cannot underestimate how important the planning is. And I think this is when it was very important to get involved with Jesper and Andreas and all the other colleagues in planning and foreseeing all kinds of small issues that we might have, so that once we start, once the cells start growing and so on, that we cannot back and then interrupt processes and stop it. So I think the planning is everything. The plans are good, but the process of planning is really the most important thing. And I really mean it. We got a lot of help from Testa Center, and they have been extremely helpful in every aspect.
Thank you, Albert. Now let's see. Yeah, so that concludes your presentation with the questions, since we just went through the last question. Thank you very much. And now I would like to hand it over to Angela. And let's see if I can spotlight you in the Teams meeting. There we go. And let's see, here we are. So Angela, welcome. You're the manager, laboratory manager at Solis BioDyne. And as I understand it, before this, you were 13 years on the University of Tartu, Estonia. Maybe you can fill in a little bit of the gaps that I left there. And then I will be happy to listen to your presentation.
Yes, hello. I will share my presentation first, but yeah, you are right. I'm at the moment working in Solis BioDyne, but before coming here, I was a scientist in University of Tartu, and also I learned chemistry in this university. But now already 8 years I'm in Solis BioDyne. At the moment I'm Chief Operating Officer of Solis BioDyne, and also take care of mostly the production and all operating processes in Solis BioDyne. Can you see the presentation at the moment? Yes?
Actually, I cannot. What about you, Jesper?
No, not at the moment.
Nothing has frozen.
Yes.
Oh, sorry, I will try.
It happened actually when you spotlighted Angela.
Okay,
yes.
let's try again.
Let's try again. I will try to.
Yes.
Yes, now it looks good.
But it went away.
No, it went away again. Sorry.
Sorry. Let's try again. I don't know what is.
Don't worry, it will work soon.
4. So.
It's what happens when you do it live.
Yes, let's try again.
Now the audience might note that we have not video recorded this in advance.
I don't know what happened.
So you have PowerPoint.
I was sure. I don't know. I shared all screens at the moment.
Yeah. So now we see it.
Now you see it.
That's good.
So, okay, but let's start. Like I already said, my name is Angela Vaasa, and at the moment I'm Chief Operating Officer of Solis BioDyne. And I will give a short overview how Testa Center helped our company to scale up production of one of our most important, or one of our, in this year, most important protein, RNase inhibitor. A little bit about our company. Our company is a small Estonian company, biotechnology company. It is established in 1995, so it is already 26 years old. We are producing PCR reagent for analyzing RNA, DNA, and for today in our product portfolio we have over 70 catalog products and many customized solutions. We have over 500 clients all over the world. You can see in the map that all these blue areas, these are places where our clients are. Our products, all our proteins are containing unique patented tag technology, which gives the product stability at room temperature at least one month. This advantage is very important because we can send our products without dry ice, and also it is very easy to use in lab condition. You don't need to set up your experiment in ice, and also it's not a problem to leave your reagent to the lab table and nothing happened. So this is very convenient for setting up also the reactions. Like I already said that we are producing several products for DNA and RNA analysis. During this year, when the COVID started, then the most important or top of our product is definitely one-step COVID kit, which what we have already sold, I think almost 8 million or even more reaction for detection of SARS-CoV-2 viruses all over the world. This product has multiplied our revenue a lot, but also this product pointed at us that what is the difficulties in production. In February 2020, when the COVID or the SARS-CoV virus arrived to Europe, and the demand of one-step mixes arrived drastically, we understood that to satisfy all these clients' needs, we need to increase our production and make this more efficient. We need to scale up our fermentation processes and also the purification of the proteins. And what we did, we actually contacted to Cytiva because we had a good cooperation with Cytiva already from 2014 when we bought first of our ActaPure system and several columns from Cytiva. And we asked that maybe we have to buy the new and more powerful chromatographs for purification. But they explained that maybe before we're going to buy something, we would like to test and find the best solution and compatible solution for our proteins, the purification. And they lead to the Testa Center. We think about that, and then we contacted Jesper, and so it started. First aim of the Testa Center was to just go and scale up the production of one of our proteins, this RNase inhibitor. But then when we arrived to the Testa, then actually, as we saw what are the possibilities in Testa Center, we thought that maybe we have to not only try to multiply, but also go through all the processes what we have so far had. So we started so, and what we did in January 2021, we put together our team, packed our things, and went to the Testa, to the Sweden. But as it was quite difficult times, it was this COVID time, and the traveling wasn't so easy. So we have to use this, actually the cargo ship, for going to the Testa. So it was quite big challenge for us to go also to the Testa Center. But we was there, and actually we was there 5 nice weeks. It was very like hard working and busy weeks. So it was quite tough time for our team. But to understand better what we did and how the Testa helped us, I will give a short overview what we had before Testa Center. Actually, we have already moved to the bioreactor, but in Tartu we have a 25-liter bioreactor. So we have to use it, but we still wanted to see what are possible ways to more increase this fermentation process. But at the moment, we had in Tartu we had the media preparation was in small quantities. We autoclave it like already said, that one way to make this media clear for viruses or other impurities is one way is to heating, or another way is the filtration. So far we had used only the autoclaving, but it takes more time, and also you need to store the media somewhere before using. Also, many purification step was still in manual mode. So in this picture you can see that here is purification of one protein by metal affinity columns. But as it was manual, then we have to use hundreds and hundreds of columns. It was very time consuming, and a lot of effort from employees need to do it for doing it manually. And also, like I said, that we have in-house already these ActaPure chromatographs, but still this is not the big scale purification. For that ActaPure 25, we can only implement the small bed size columns. So we had all these systems, but they were quite not small scale, but still meet scale of production and purification. And if to compare, then one of this cycle for production and purification took almost 3 or a little bit more, even months. When we arrived at the Testa Center, then like I already said, that first was to just go and try to produce something. But when we arrived there and saw that there is so many different possibilities, we saw that maybe it's good to go with all these processes step by step and look what are the places we can increase or make it more efficient. So we started to optimize our fermentation process. And as Testa Center has many bioreactors, small bioreactors, so it was very convenient to do it, because we use 6 small bioreactors simultaneously. We optimized all media composition, pH, oxygen level, temperature profile. It was very fast, because all these conditions we can, like you can imagine, 6 reactors, you can already start changing 6 different parameters. So it was very easy and fast to optimize these conditions. Then when we was almost agreed that these conditions are the best for our proteins, we moved to the 50 liters bioreactor. It looked a little bit scary at the beginning, because as you can see in the picture, there is a lot of like tubing. Control panel was lot of buttons to what to push, but actually it was very, very convenient and easy to use. Also, in Testa Center, we tried first time to filtrate the media. Here you can see also that 50 liters media, we just mixed all components in one bucket and then just filtrated to the bioreactor. It was very nice to work with bioreactor, but of course we understood that actually to get the best result, you have to know very well your behavior of your cells, of your proteins, and it's not so easy that you just scale up. So you have to control all details very, very, very well. And with this bioreactor, it was easy to do. Of course, when we already produced a lot of cell mass, the next step was the purification and harvesting the cells. Also in these steps, we took several new methods and tried several new methods as Testa has several alternative equipment and methods for doing it. So we tried for harvesting the cells, we tried several centrifugation steps, DFF systems for filtrations, and so on. Also, like already Jesper mentioned, that in Testa Center there are very many different chromatographic options. We used ActaPure 150 and ActaPilot. This was already scaled up a lot in our at the moment what we use. Also, we had chance to use very big columns. We had chance to pack them ourselves. So the biggest column what we used, that was 1 liter media inside. Actually, there, as you can see also the picture, that there will be the options to make much bigger ones. But in our situation, the 1 liter was already quite big. Also, as Cytiva has very many different medias for purification, then it was very easy also in Testa Center to use or try several ones. So we had chance to try different metal affinity chromatographic resins, different ion exchange resins, and also dissolving media and dissolving columns for optimizing our purification. So, and like I said, that the good thing was that we learned how to pack the columns ourselves. So far, we just buy the pre-packed one, but now we have the knowledge how to pack ourselves, make the columns as big as we need. Of course, everything was possible because of this very supportive team of Testa Center. So. Like already Albert said, that in the beginning when the planning started, they was very supportive, they was very detailed, they asked a question and put us to think about what to do, how to do, and does it enough for this time. And also there was the ideas and the flexibility of Testa Center and the team. For example, the customized solution. In this picture, you can see even they didn't have the right or maybe this kind of equipment what we needed, but still they find the solution. In this picture, you can see this DFF customized solution of the DFF filtration. So, but it worked very well. It seemed that a lot of tubing and flask, but it was very nice and helped us a lot. And of course, there was also the possibility to see and meet other partners of the Testa Center. We had possibility to see depth filtration system from PAL Corporation, also the continuous centrifugation from Alfa Laval. So we experienced a lot different methods, different applications, different ways to do this process more efficient. And now, after that, we came back to the Tartu. We haven't implemented all knowledge what we got from the Testa Center, but the situation at the moment is that we are planning to buy a new bioreactor, and even maybe not this 50-liter one, but already the 200-liter ones, because these needs for production increased still. We have already upscaled our DFF system for clarification of the lysate. We have bought 6 ActaPure systems for simultaneous purification, and also new ActaPilot 600 system. And of course, larger columns. These columns what we can pack ourselves, and various media to optimize more or make the columns like we like. All these knowledges, this implementation has already increased our production or make it more efficient already 30%. But I think if we can implement all these knowledges and these methods what we got from the Testa Center, I think we can increase it even more.
So, Angela,
so it was very nice. Sorry.
I noticed that we're coming up on time, so I would like to encourage all the audience.
Actually, it was the last slide. Next one is already the thanks slide. So
I just want to encourage the audience to ask the questions that you have to Angela now, so that you have time to write them down and send them in, so I can pick them up and forward them to Angela. Thank you. Please continue, Angela.
Yes, actually, that is only the thank you slide, because it was very great pleasure to be in Testa Center. This opportunity opens our eyes a lot, and now we know if we would like to do something or we would like to go further, is good if you can try it before you're going to buy something. So then you can find the best solution and mostly compatible solution for your applications. Thank you.
Thank you. Thank you, Angela. Great story. I'm glad to hear it, and I'm happy to give you the questions that have come in during your talk. Let's see. So I think we have a couple of students who want to work for you now, because the first question is, are you looking for thesis students?
Actually, like I already explained, that we have grown a lot. We have widened in any areas. We have widened in our production side, but also in people side. So actually, we are looking very good and talented lab people, and also as in our companies, not only the lab, but also the sales, marketing, and other parts. So actually, we widen a lot, and good people are also time welcome to our company.
I think we have an academic audience, because the next audience is also somewhat related. So it's like, so the question is Solis BioDyne a university spin-out company, or do you have any other similar collaborations with the academy?
Actually, we are not the spin-off at the university, but still, as a lot of our employees comes from universities, so we have very contacted to universities. So, but also we are doing cooperation with mostly the University of Tartu, but also all over the world. As our clients are many clients are universities, so there is cooperation and with many universities.
Okay. And the third question would be, and the last question is, your project seemed to be exploratory. Was this intended from the beginning, or was it not? Yes. Did you have a very detailed planning, and then you ended up in something else, or?
Yes, like I mentioned, that actually first we just started to look around to find possible partners where we can just increase or scale up our production. But in Testa Center, as there was so many different and various equipment possibilities to try different things, then we realized maybe it's not that it's a good time to take this this time for optimization, because at home, when you run your processes, you usually do it like you do, but and you have your regular equipment. But in Testa Center, we had chance to try different ones. So we planned to go just for production, but actually in Testa Center, we realized that maybe we need to optimize more and look what is the possible processes we can increase. Maybe it's not only the fermentation, but it is maybe, like I already said, that maybe it's a bit with this purification step or centrifugation step, or actually we go through all steps and find what is the main limitation and go through this one. So it was, yeah, the aim was first one, but actually we got more we expected.
I'm glad you did. Happy to hear. So with that, the questions for you, Angela, are finished, and now we have a new question on the Mentimeter for you in the audience to be able to ask us anything, really. So just shoot a question there. Nothing is a too small question. And while you're doing that, I have two questions that I prepared, one for Albert and one for Angela. So I'll start with you, Albert. Let's see if you are there. Yes. If you're interested in running a project in the Testa Center as a student or as a researcher, how do you get started?
I think you would need to have a very clear idea about your goals. What are you trying to achieve, and what would be the added value to your project when going to Testa Center? Of course, if you have been there, it's a lot easier to understand what you will be gaining. And I think there is so much at Testa Center to use and test and to understand. It's really immersing in the real life environment. But I would also suggest that you keep it simple, straightforward, because the complexity grows. There are a lot of things that you may not predict, and when you come in this environment, that is very difficult to foresee. So have a very clear goal and keep it simple. I think that would be my suggestion, actually. And then you will get super support. You will be in the right kind of environment. You will get access to most advanced bioprocessing equipment that is available.
Very nice. I bet for the Uppsala University also, I know there is coordinators working with you specifically, right?
Once again,
so there are one or two coordinators that you can reach out to, or yourself, yes, perhaps.
Absolutely. There is I think what needs to be mentioned, that Testa Center is viewed from the Uppsala University's side as a very important link to the industry and the possibility for collaboration. There is a steering group at Uppsala University that combines coordinators from medical pharmaceutical faculty and the technology and engineering faculties. And as it has been mentioned in one of the questions, Uppsala University has allotted 25 weeks at Testa Center. And these weeks can be used for education, for research projects or innovation. That is associated with the researchers at Uppsala University. And this has been enabled by direct support from the rector and the faculties. So 25 weeks a year, that's, in one of the labs at Testa Center. This is a fantastic opportunity.
I'm very glad every time I get someone from Uppsala University to visit. I hope to see you soon again. So Angela, I have a question for you as well, and I got interested about this question of collaborating with the university and how your company might, how the collaboration between the university and your company and also the Testa Center, how that makes you push forward, and if there is like a synergies between these entities. Do you have any reflections on that?
About this, sorry.
perhaps from your own story.
Actually, the cooperation with, like I explained, that for the universities we have, as the product what we produce are used by universities, by scientists, actually also the diagnostic companies, kit makers. So to understand these the needs for different clients is very important, and that's why the cooperation is also very valuable for us to understand how to use our products, how we can improve our products. In Testa Center, of course, the cooperation between the Testa Center, I don't know what you can get from our company from in cooperation mode, but for us it's very like important this kind of possibilities. If we are, as I explained, that we are still small company, and then we need the support or possibility ways to test before to do ourselves. So the cooperation between these kind of testing centers for small companies are very, very valuable for our companies. And also the knowledge what we got, because in Testa Center you have also, as you have several different projects in your house, so the knowledge what you can share for us is something maybe we cannot get anywhere.
I'm glad to hear. And then for Jesper, on that note that Angela asked us almost, like, what is it? What's in for you? What's in it for Testa Center? What's in it for Cytiva? Maybe you can take us away with that, since we have answered all the questions in the chat. I think this is a good way to end, perhaps. Maybe you can say something about that, why we do this.
Yes, excellent. Thanks, Angela. I unmuted myself for a reason. I was hoping that I would get the chance to answer this. So we do learn a lot from you guys as well, Angela. One thing that we have in our backbone is a little bit the big pharma, the corporate way of working, which there are, of course, things that are really good with being a big corporate background, but also you can get a bit slow. And sometimes you get a little bit, how should I say, administrative. So the input that I feel we got a lot from Solis BioDyne is the agile way of working, the way of attacking a problem and going for a solution when you see it. So that type of attitude, I think, is a great way that you need to have when you're an entrepreneur, and getting that entrepreneurial spirit into the business, and also, let's say, contaminating Cytiva a little bit with that entrepreneurial spirit, I think is really good for Cytiva. We want to be as agile and customer focused as we possibly can, and getting to know our customers and the small customers better is a big thing for Cytiva, really.
Right. With that, I think we're just about on time. The questions, we're done with the questions. I would like to thank everyone that was watching in the audience. I would like to thank all the presenters. Thank you very much. I hope to see you again very soon. And until then, take care and stay out of any COVID problems out there. Cheers. Have a good time.
Thank you very much.
Thank you.
Thank you. Bye-bye.
Thank you. Bye.